Everybody. First of all, I want to thank Cesab and all the team of Cesab for organizing this meeting, this presentation, and so CESABINARS and the program of CESABINARS. Thank you very much. I also want to thank the organizers, Nicolas, Romain, and Camille, and Maud, who gave us support not only now for the Cesabinar, but also for the project along the 3 years of project we had. And I also want to thank not only Cesab, Symbiose Project, Seba, and Biota FAPESPI, and we'll have some more acknowledgements at the end. So, and I want to also tell you that we're going to divide the presentation. I'm going to speak a little bit now, then I'll pass the word on to Jerome, and then George Brown, which is also our colleague and partner in this project. He's also one of the PIs of this project. He'll be presenting some part at the end of the presentation. Okay, so thank you all, and thank you my partners, Jerome and George, for this project and our projects which we are taking along together at this moment. So I'll start talking about FaunaServices, which is the project which is a teamwork, it's a relationship between soil microfauna biodiversity and ecosystem service deliveries across land use systems in the neotropics. Next slide, please. It's mainly a very big teamwork. We had around 18 people participating from various countries: Brazil, Canada, Colombia, China, France, UK, and Germany. So those are some photographs of our meetings with workshops in Montpellier and in Piracicaba, where I am right now. So we met many times between 2022 and 2025, either in Montpellier or in Piracicaba. Next slide, please. So beginning with our presentation, what are neotropics? Well, neotropics are mainly a biogeographical region which encompasses South America, Central America, part of the Caribbean islands, and south of Mexico. But our project mainly focused in South America, Colombia, Peru, Ecuador, Venezuela, Brazil, okay. And we focus mainly in 2 biomes, but also we came in with some data from the Cerrado biome. So we worked in the Amazon biome, we worked in the Cerrado biome, and we have data from the Atlantic forest biome as well. Next slide, please. So mainly we are working in this project. The idea is to work with biodiversity, soil biodiversity, and their effect on ecosystem service delivery. So mainly global ecosystem delivery from the neotropics. We can consider mainly 3 main ecosystem services delivery, mainly carbon storage, because we have huge carbon stocks and a big climate relevance in this region. We have great amounts of water in this region as well. So water regulation is also a very big ecosystem service we have in this region. So hydrology and drought resilience across biomes is one of the objectives of main lines of our studies. And also it's a very big biodiversity hotspot, exceptional above and below ground diversity. So mainly these three ecosystem services, these 3 main lines of ecosystem service were what we're mainly approaching in this project. We're approaching this project and the projects we are coordinating at this moment. Next slide. So mainly these biomes are under threat, mainly by 2 drivers, climate change and land conversion. So those 2 drivers are producing a sort of loss of biodiversity, water stress, water deregulation, affecting regulation and deregulation of water. Also that affecting water stress, also fire. We have that comes to vegetation change, but we also have losses in carbon and nutrients. So mainly we have a big threat according in the neotropics and in these biomes due to these 2 drivers, which mainly affect biodiversity, which is one of the ecosystem services we're studying, water regulation, and carbon and nutrient losses. So mainly our main idea in this project was to understand this climate change and land conversion and its influences on carbon and nutrient losses, water degradation, and biodiversity loss. Okay, next slide. So land conversion in the neotropics and mainly in the region of South America, which we studied, is a major threat. Here on the left side of the screen, we can see an evolution in the last 20 years of how deforestation has advanced, okay, in Amazonia and Cerrado, having big influences mainly in the Cerrado and Amazonia. On the right side, we see also the effect on the Atlantic forest, the original area occupied by Atlantic forest, and nowadays the remaining native vegetation in the Atlantic forest. So mainly these three biomes have been very affected during the last, not only 20 years, but the last century, by human occupation, agriculture, and other land uses. So mainly we had a very big land conversion in these areas. And it's, as we say, it's a major threat, okay, to soil biodiversity and other issues related to soil biodiversity. Next slide, please. So mainly water stress, which is one of the main issues we have been studying in this project. Through global warming, we are affecting regional rainfall conditions. These regional rainfall conditions affected by global warming will affect water stress, water holding capacity in the soil, water storage in the soil mainly, which can affect forest loss. So less water in the soil, forest changes, forest loss, or forest conversion as well. So mainly we have, or it could go on to deforestation as well. So we have an accumulated deforestation, which feeds back into regional rainfall conditions. So mainly it's a loop, global warming, regional rainfall conditions, water stress, forest loss, more deforestation, less rainfall, etc. So we are looping all the time, and we are mainly transitioning to more degraded conditions and obviously changes from systems, mainly as we see on the right side, where some areas in the Amazonia are stable, some areas are transforming forest to savanna. Sometimes we have some biosystems, two systems which live next to each other. So we have forest and savanna, but it can be in disequilibrium. So we can transform savanna to this bisystem, or this bisystem to savanna or forest. So this paper, Critical Transitions in the Amazon Forest System, we see how different areas of the Amazonia are changing or are savannizing or transforming to savannas, or from forest to bisystems. So bisystems mean savanna and forest together. So we see that really water stress and can bring great changes in the Amazonia and the other ecosystems or the other biomes we're studying as well. Okay, next slide, please. So what about soil biodiversity in all that? So how does soil biodiversity come into all that? So next slide, please. So mainly when we see the role of soil microfauna biodiversity, mainly through land conversion, climate change, so that will bring soil biodiversity loss, which will affect soil properties. These soil properties, mainly soil structure, so soil nutrients, soil porosity, can affect water stress, water regulation, less water in the soil, or less water storage in the soil, so less water supplied to the plants, for example. That can be the soil properties also can affect the carbon and nutrient budgets in the soil, so less carbon, loss of carbon, or eventually higher amounts of carbon, which we know we have to see that budget, nutrient loss or nutrient gains. So mainly we're talking about degradation, so mainly it will be mostly losses. So the changes, soil biodiversity will affect especially soil properties, physical, chemical properties, which mainly affect 2 points in this figure: water regulation and carbon and nutrient budgets. Okay. Next slide, please. So the objectives of FaunaServices mainly was to assess ecosystem services delivered by below-ground microfauna in neotropics. For this, we identified environmental drivers. We identified and obtained data or constructed a big database identifying soil microfauna communities. We also addressed soil functioning through big databases of soil, where we have the influence of organic micro decomposition, nutrient cycle, and soil structure. So mainly we try to put together or analyze together soil microfauna communities, soil functioning, because we have this data. And apart from that, we evaluate ecosystem service, so carbon sequestration, nutrient availability, water availability. And at the end, we end up with nutrient budgets, mappings, and other data analysis, which we're doing now, trying to assess all this, crossing soil data, soil biodiversity data, microfauna data, and ecosystem services data. Okay, next slide. So what is soil microfauna? Soil microfauna is mainly a big, enormous array of taxas, which are listed there. I'm not going to go into all the details. So many microfauna is a big array of taxa that live in the soil and that will modify the soil or will live in the soil, and through its living, eating, will modify the soil, will use soil as its habitat and affect its structure and other nutrients and carbon. So mainly microfauna will be there living in the soil and bringing, changing soil properties and affecting soil properties. Next slide. So in this case, in FaunaServices, following the objectives, we built a large-scale synthesis, which was the objective of this project of the Cesab and NSF and the FRB, sorry, and PAPESP and CNPq project was a big synthesis, mainly on 4 harmonized databases. So we built a very big soil microfauna database based on the TSPF standardized protocols, where we measured, or we have in that data, we collected in that database abundance and biomass for around 40 taxa in 3,500 sites. We built a very big earthworm database with quantitative and microhabitat records around 3,000 sites and with around 700 species in this database. We have the soil profile database, which I'll be describing in a few minutes, but it's a great soil characterization across the region, across the neotropics in South America, with more than, he says 5,000, we have more than 5,000 sites, we have around 12,000 sites. And we have the effects on ecosystem engineering, which we also have biostructure versus controlled soils. We have in the database around more than 12,000 observations. So mainly we have a very big database on these 4 harmonized databases. This is mainly the great product of the FaunaServices project, was the building these 4 great or big databases of microfauna, earthworm, soil profile, and ecosystem engineering effects. Okay, next slide. So I'll pass the word on to Jerome now. He will explain the different databases, and we'll come back later on. Thank you.
Okay, so like Miguel said, we built 4 databases, and it took us some time. It was quite an effort. So and it was quite difficult actually. So I will present a bit how we did and what we succeeded to do. So first, the soil macrofauna database. So it's a database that gathers data of soil macrofauna abundance. So it can be either the number of individuals or the biomass of roughly 40 taxa. And all these taxa, they were sampled in a kind of standard way, which is basically taking monoliths of soil, like you can see here. So usually for one site, people would take 5 samples, 5 monoliths. So you have a picture here, and then they would extract manually the soil fauna. So we gather data that were sampled with this with method, and they also come with associated data like vegetation cover and soil properties. So to do that, one of important step on it was really useful. It was to build a standard template, so people could format their data to fit in the database. And what was really useful is to put it online, so it's on Zenodo, so everyone can use it. And so people are starting to use it for their own project, and it's making a kind of a collective effort. And it's working much more than what we expected. So one difficulty is to standardize the data because, so there is an official standard, but every team will modify it a bit to fit its own logistics. And so we needed to harmonize, standardize all the data. So that took also a lot of time. So we didn't expect it to be so complex actually. So that here is just one example where we try to rescale all abundance to a similar depth, because some teams, they would sample only at 10 centimeter depth, for instance, and other would go up to 40 centimeter deep. So it's hard to compare directly the numbers. So we need to rescale a bit the numbers to have the same volume, same sampling effort, basically. So we did an effort on that, and that's just an example to show the pathway. We also made a Shiny application. It's a kind of a web interface where people can drop their data sets, and then there are some kind of automatic checks to see if the data are in the right standard, if they are missing data, and that kind of things. We also published a book to explain what is macrofauna, basically, how to sample it, how to identify it. And so far, it's in Portuguese, and we will translate it in Spanish and in English, because it's mostly focused for South America. So what did we achieve? So we succeeded in gathering data from 3,500 sites, so across the neotropics, I would say. And they're mostly from Amazonia and for Atlantic forest, which is called Mata Atlantica. And there are much less data on Cerrado. So we decided to start to publish the data. And that was a bit a challenge, I mean, in the sense how to publish that, because it comes from an aggregation of data sets. And we wanted to keep the identity of the original data sets instead of publishing a whole database that we will do later. But for the moment, what we decided to do is to publish dataset by datasets. So there is a kind of collection in GBIF where you can access to the original datasets. It's not all published, but we are doing it. So that was for the macrofauna database. So now there is a earthworm database. So the earthworm database, basically it's the same sampling protocol on the left, which is quantitative. And there are also a complementary sampling, which is based on a microhabitat sampling. So typically logs or bromeliads or piles of leaves or that kind of things. And regarding, so it's other sites. So some sites are common with the first one, but some are completely different. And we succeeded to compile 300 sites, 3,000 sites, and roughly 700 species, and most of them are not described actually. So we have barcodes and that kind of stuff, but there's still a lot of work to do to have the name of the species and to describe the species. After we have the third database, which is about soil profile. So I will let Miguel introduce this database.
Okay, back again. So the soil profile database was mainly constructed based on two basic databases. One of the basic databases we use was the soil data collected in the sampling points of the soil macrofauna and soil earthworm sites or locations. So many of those soil macrofauna and soil earthworm data came accompanied with soil data, normally until the depth of the sampling, some 20, 30 centimeters, sometimes 40 centimeters. So that was one of the soil databases we used. And the other soil database we used were regional databases or national databases from Brazil, from Colombia, from Peru, from French Guiana, and well, that's still growing because we are still collecting data. But anyway, until this moment, we have data from those national or regional databases, which are mainly come from soil service, so from national or regional soil services, which are based on soil profiles. So once again, as Jerome said, we can go on to the next slide. Jerome, please. We have more than 8,000 sites. It's a bit larger, that number, joining the national and regional soil databases coming from soil service surveys and the soil macrofauna and earthworm soil data. We have around 12,000 sites, more or less. We also had a very big issue for standardizing this data because we had different depths also of sampling, but we also had a very, very, very big job because we had different units. The soil properties and the soil characteristics came in different units. So we had to standardize this unit. That gave us a lot of work. But eventually we managed to standardize those units to be able to compare and the soil data, especially the soil physical and chemical data, which we extracted from soil database. So that's the map of the region which our project covers, and all the sites where we have soil data. It's mainly soil chemical data, so we have carbon, phosphorus, nitrogen, calcium, potassium, magnesium, pH, okay, soil base saturation, sum of bases, and we also have physical data as, for example, sand, clay, silt, bulk density, and some profiles. And apart from this data, we also calculated using some modeling. So we started doing some modeling with these soil profile databases, using what we call pedotransfer functions. We, as one of our ecosystem services, we're going to study is water regulation. So we started calculating already water availability in the soil, water storage in the soil, using these models called pedotransfer functions. So in this soil profile database, we already have data on soil water availability, retention, water conductivity, hydraulic conductivity, etc. So mainly that's that. So we had a very big, we used a lot of time homogenizing and already modeling some data, especially related to water regulation. Next slide, Joe. I pass on to Jerome now.
Okay, so I will talk about the last database. So this one is about the effect of the ecosystem engineers on soil properties and ecosystem functioning, actually. So the ecosystem engineers are termites, ants, and earthworms. And the database consists of data about the properties within the biostructures and outside the biostructures. So we can compare them and compute effect size, basically. So we gather data a bit everywhere, actually, because we were a bit more than South America. And so we have now 12,000 observations. So that's what took really the most of time, building all this data. It was really long and difficult, but so we start to analyze the data now. And so I will show you a few results. So the first result is just simply to have an idea of the biodiversity in soil, because we all know that Amazonia for Atlantic forest are really like highly diverse in terms of plants and above ground animals. But what about soil fauna? So we did analysis with earthworms data set. So that's just a rarefication curve, but we can see that if we take all the samples, we reach and we extrapolate it to 1 kilometer, so just 1 kilometer, we reach roughly 700 species. So it's really a local estimation. So you can imagine if you scale up that to the global biome, that's what we're doing at the moment. With the number are so frightening that we are still not sure about what they really mean, and we prefer to take our precaution before showing them. But it's really overwhelming. I mean, the number is really crazy. But that's soil biodiversity. It's what it is. So we also did the same thing to compare Amazonia and Atlantic forest, just to see who is more diverse. And here we found that Amazonia was more diverse compared to Atlantic forest for a similar sampling effort, which was not what we expected because Atlantic forest, it's a hotspot of biodiversity. So we were expecting the reverse, but actually it doesn't seem true for worms, actually, at least. So another topic we started to investigate is the invasion by exotic worms. Because you might know there are like many species of worms, and some, of course, are native, so they are from the place, and some are just new ones, new species. And oftentimes, these invading species have a strong effect on soil properties and ecosystem functioning. So it's important to know how much invasion occur and where to be able to anticipate a bit the consequences. So here in this figure, you have the relative abundance, so native, exotic over native species. So basically, when you are here at 0, it means that you only have native species, native individuals. Sorry, because it's abundance here. And let's say here on 1, it means that you have 100% of exotic individuals. Okay, so that's an histogram of the ratio across the sites. And what you can see is that globally in the natural places here, it's mostly dominated by native species, but you also have in some cases exotic species. So it means that even in the pristine areas, you can find exotic species. And when you go to more like anthropized situations, then like expected, of course, you have more and more exotic individuals. Okay, so the communities are becoming dominated by exotic species. After we're wondering what are the consequences of land use conversion, sorry, on soil macrofauna communities this time. So does it change basically the composition of communities, or do they stay similar? So what we do is we simply plotted the communities, so 3,500 communities here on the PCA. And so it's not a constrained analysis, it's just really the rough PCA. And what you can see is that there is really a natural and strong opposition between, on the left, in pink, all the tree covered areas, so it's mostly primary forest and some tree plantations. And on the right, it's a new land use type, like crops and grasslands. And you can see there is a strong difference in composition, in tax sector composition between the two type of land use. So that's quite a strong result. And regarding the effect of ecosystem engineers, so this time we focused only on 3 taxa, and we look at how the different soil properties differ within and outside their biostructure. And we looked at many properties, soil properties and ecosystem properties. Here, ecosystem properties are not shown. We are just focused on soil properties. And you can see that in most cases, the nutrients are higher in the biostructure, they're higher than in the control soil. And also many things related to physical properties of the soil. So we still didn't do it, but with this information afterwards, we can compute what Miguel's saying. We can compute soil functioning with pedo transfer functions. So we are doing it at the moment. So perspective. Miguel, I'll let you talk.
Okay, thank you, Jerome. So one of the first perspective we have of this project and the use of the soil of this big database, soil microfauna, the 4 databases, is to link soil microfauna communities to soil functioning, as Jerome just said and I said a few minutes ago. So mainly we have around 12,000 sites of soil data, chemical and physical data, and many sites of soil fauna data. So the idea is to combine this data and see what relationship we can find, mainly the Mata Atlântica, Cerrado, and the Amazon biomes. So the idea is to try, we're already working on this through our PhD students to understand the relations between the soil microfauna we have in our database, the soil physical and hydraulic properties, and land use change, okay? And how these interactions or these relationships will interact with ecosystem services, mainly looking at 2 main ecosystem services related to the soil data, which are water regulation and water availability. The idea is to have direct relations between and interactions between these attributes we're analyzing. The idea is to work also with some modeling, okay, not only through pedotransfer functions, but also through hydrological modeling to be able to understand how these interactions between soil microfauna, land use, and soil physical and hydraulic properties affect water regulation and water availability. Okay, so mainly that's one of our perspectives which we are already working on at this moment. I'll pass the word on to now George.
So thank you, Miguel and Jerome. One of the additional services that we were interested in has to do with impacts on nutrients and eventually on plant growth. So what we did manage to do is map and show earthworm abundance across the different biomes here, as you see on the left-hand side. And then we took a meta-analysis which had been produced by Steve Fonte a few years ago regarding impacts of earthworms on plant production, particularly grain crops such as legume grains like soybean or grass grains like maize, wheat, rice, etc. And then we took some data regarding some of the most common and most abundant earthworms in Latin America, particularly in Brazil, like Pontoscolex corecrurus, and some data regarding the impact of these earthworms, this earthworm species, on nitrogen availability in casts and phosphorus availability in casts. Next slide. And then we took the annual crop coverage of Brazil, which represents around 7%, of which probably the largest surface is regarding maize and soybean. Many of these crops are grown sequentially, so you can grow up to 3 crops in the same plot of land, and this is shown here in terms of the amount of in the lower left-hand side, which just shows that we have several crops growing on the same plot of land over the year. So that covers around 60 million hectares in Brazil. And then the pastures, which cover around 160 million hectares, which represents almost 20% of Brazil's surface cover. So next slide. So what we did is we took the land use area for pastures, soybeans, and maize, and the annual production data, how many bushels per hectare, tons per hectare those crops were produced, and how much the annual market for pasture grass would represent in the country of Brazil. And we took the standard rate of impact on production by earthworms taken by the Steve Fontes and Jan Willem van Groningen's meta-analyses. And we calculated the percent contribution of earthworms to pastures and both grain crops. And you can see there in the table that annually that represents close to 20 billion in terms of impacts on pastures, and around 2 billion US per year for both soybean and maize. And then we calculated based on the average biomass of earthworms in those fields and both pastures and grasses, and how much carbon, how much nitrogen and phosphorus were made available by the castings produced per year in those areas. And this is then the value in fertilizer equivalents that is provided by earthworm casting in those soils. So in the pastures, it represents about 20 billion US per year in nitrogen, and around close to 300 million US dollars in phosphorus. And for the soybean and the maize, well, close to $1 to $2 million in nitrogen, and around 11 to 20, $1 to $2 billion dollars of nitrogen, and around 10 to 20 million US dollars of phosphorus. So that's a pretty important contribution. These are the first time this has been done. Of course, there are many caveats regarding this, but it just is a preliminary estimate of earthworm contributions, both to nutrient and plant production in Brazil. Next, thank you.
Okay, so I think it's my turn to conclude. So overall, the project, I would say it was really interesting and really a lot of work, and we are quite happy of what we have been able to do. But we see it like a start actually, because we still have a lot of things to do, and it's really gave us the opportunity to grow our network. So, for instance, during the project, we had people from Canada joining us, sorry, through a joint call between Canada and Cesab, and it really helped us to enlarge the topic, and including food webs models in the work. And it also allowed us to get new fundings for different projects. Like Miguel said, we have a joint PhD program with CNRS on Brazil, and different other projects. So it was really helpful to have this funding. So in one word, thank you to the founders a lot, and we are welcome to have questions.
Thanks a lot, Jérôme, Miguel, and George, for this very interesting presentation. So we have now time for questions. If you have questions, you can raise your hands, or you can write it in the chat. I encourage you to unmute yourself and turn on the video for asking your questions, so it's more interactive somehow. There's no stupid question, so don't be shy. It's really time to get to know more about the research that has been presented. I don't know if there are any questions among the participants. I might have a very simple, stupid question first to break the ice. I know that the tropical Atlantic forest and the Amazons have kind of different species. I was wondering, you were comparing the earthworm communities. Are they the same earthworm, like these 200 species you find in the Mata Atlantica and the 700 you find in the Amazon? Do they share, are they the same, or do you have speciations among them?
Okay, so I will
Just
start
out of curiosity.
George will complete if needed. Yes and no. So first, what is really striking is that most species occur only in one place. That's really crazy, but that's native species rarely occur in more than 5 places. So and from this point of view, it's completely different biomes, but there are exotic species which are common to biomes. So I don't know if George, you want to complete.
That's correct. Perfect.
Yeah. So Carlos, if you have a question, you can ask it directly.
Well, thank you for the presentation. You're getting very interesting results. And my questions goes towards if you have any explanation so far, or what are the lines that you are thinking to explain the fact that the Atlantic forest has a lower diversity than the Amazon forest. I know it's not easy, Miguel, but what are your earlier thoughts on that?
George, should I make a tentative reply?
All right, and if you need, I fill in.
Yeah, so I think I think there are maybe several explanations, but one is so first one thing that goes in your sense is that it's even more surprising because it's roughly more colder. I mean, part of the Atlantic forest is colder than the Amazon, and regarding that, we would expect even more species based on Ellen Phillips' science paper in 2019. So it improves even the surprise. It increases the surprise. And maybe what I was thinking, maybe it's because actually most of the Atlantic forest is already cut. And I was wondering if that's a reason. But I mean, if it's true that if we compare, when we compare the rarefaction curve, it should not, I mean, it should still be higher in the Atlantic forest. But yeah, for that, I don't have any. I have an explanation for the gamma diversity, but not for the local diversity. Maybe George has a better answer.
Yes, one of the interesting things we also saw in our data set is that there's a positive relationship with rainfall and biomass, and so it seems that the Amazonian rainforest supports, in general, a higher earthworm biomass due to more, higher rainfall. Well, kind of overall what we had seen in terms of the data so far seem to show, okay, there's probably a relationship with climate and for sure land use history. And you have in the Atlantic forest immense, very long lasting history of perturbation and deforestation, which is just a lot more recent in the Amazon, at least in terms of impact of the perturbations. Whereas in the Amazon, you have perturbations which are at much smaller scale, and even though they also have a very long history, that impact overall, for instance, of the Amerindians over time was much smaller. So there's both the impact of land use history in the region, the historical importance of that, the level of impact on the soil and on the vegetation, and some climate impact factors which are still not well resolved.
Can I kind of put in a grain? I think also, I don't know, we have to see the sites that were samples, but probably also in the Atlantic forest, we have lots of restoration projects and regeneration projects, which brings lots of ages, different ages of different types, if it's a pioneer forest or a secondary forest or a more stable forest, and eventually the age of the forest can also be, so that a bit in the line that George just said, the land use history, and if we have sites in restoration areas, because we know that the Atlantic forest grew a lot in the last few years due to restoration projects. So probably that, and also, which is work we're doing together with not only soil fauna, but with soil vegetation, the relationship between vegetation restoration and soil degradation. Because I always defend that the soil we have nowadays is not the same soil that was there when the forest was cut down. So the soil is totally different. So that soil degraded, that's been restored, or that forest has been regenerated, especially in the Atlantic forest, is very different. So that can also affect the soil fauna biodiversity.
Yeah, there are almost known pristine areas in the Atlantic forest, and so that may explain it. Now, thank you.
So I see a question in the chat from Marius Menchek. I don't know if you've seen it. He says, regarding the results George presented here, are you planning to look at the factors increasing, reducing the contribution of earthworms to the monetary values? For example, the effect specific management strategies or intensity, fertilization, pesticide, etc.
Yes, for sure. I think we have a lot of data showing difference, particularly in tillage management, crops that have been tilled versus crops that have been no-tilled. And of course, that's very different in terms of the abundance of macrofauna in general, but particularly of earthworms. So there's definitely enough data for that. In terms of management intensity strategies for fertilizers and pesticides, not so much. I think almost all of our land uses, particularly in the Atlantic forest region, are all fertilized and receive some kind of pesticide, be it herbicide, fungicide, or insecticide. So having the comparison between like more organic or agroecologic and normal agriculture, we just don't have enough data for that. But of course, we do have enough data to say that there are differences of impact that these things impact the earthworm community. So indirectly, we could believe that yes, as soon as whatever you do that will end up having more earthworms will have more impact monetarily on the contributions to ecosystem services. So.
Marius said, could be also interesting to compare sites that differ in their time after land use conversion. I don't know if that's kind of data you have access to.
Yes, for sure. Time series data is also something that's quite rare overall, and I think all of us know that very few people have done monitoring just as well as in Europe, where people started looking at earthworms 120 years ago, and here we've only just basically started in the last 40 years. So yeah, I think certainly time after conversion in terms of taking sites that have this, like Miguel says, this regeneration process or management over time, finding those sites on the same soil type, in the same climate type, so that they're comparable is always a big challenge. And when you use agricultural field experiments, of course, it's great, but you're always limited by your ability to scale up because the field stations don't always represent the reality of farmers. So we have been doing this with no tillage sites in Brazil. And yes, we see that there definitely is the chance of using the data that we have gathered on different ages of no-till to look at the impacts in ecosystem service delivery. So that's one of the things that we'll be exploring also in our data set.
Thanks. Any other question from the audience? Else, I still have questions a bit linked to the answer of Jérôme. I find it very impressive the 4 large database that you compiled. Like 8,000 sites for soil, 3,500 sites for macrofauna. But knowing the size of Brazil and the neotropics is huge, right? So it might be little. So I don't exactly know the amount of small-scale spatial variation. So with such database, can you make reliable maps? I've seen that, George, you shared a map of earthworm abundance. So it seems to be possible, but is it so? Do you have enough data to make predictions reliable and to extrapolate over the whole Mata Atlantica, the whole Amazon? And which one is easier to predict? Is it soil macrofauna easier to predict versus soil profile? Or if you could share a bit your insight from this.
Yeah, no, we never have enough data to put it short, but it's already so much work to have that. But of course, I mean, we can make maps. That's not an issue, even with 2 points, as you know. But you know, the real question is the quality and like you mentioned, and that's really hard to. It's really hard to assess actually. And so at the time we do our best, we are pretty sure. I mean, how to say, pretty conscious of the limits of the effort of the exercise. And I mean, we're still, how to say, we're still in the process of doing these maps. And what we want to do is to filter basically and to remove places where uncertainty is too high. That's how we want to approach it. And but the thing is, you know, Amazona is is so difficult to go in and to have new data. So it's really a challenge. But yeah, I mean, the only thing we can do is make the map of uncertainty be clear on the limits, and that's it. But for the soil profile, I mean, I'm more confident because we have much more data. But for the macrofauna, and it also depends on the feature of the fauna you're talking about. Because, for instance, abundance is really the harder to model. That's just because the distribution of the data is really weird. But if you look at the species richness, for instance, it's much more robust. Or text presence absence, it's really easier. So it depends on what you're looking at.
Even if you seems to say that there was earthworms that were very specific to a certain valley or a certain site, even with this, you can predict kind of the community richness somehow more confidently
because
based on.
we don't model species by species.
Exactly. Okay, cool.
Yeah, because it's a typical case. You have too few data to fit a model, and you have 2 points. So, okay.
Just adding up to that, as we have lots of the macrofauna data, sometimes it's clustered. So it's probably difficult to have an overall model for the whole region. But probably we can build models for simple clusters, combining soil data, soil microfauna, and see what happens at a regional base and not on the whole neotropics eventually. So, and then we can get the specifics in a little bit more.
A last question from the chat. We'll call it a session. Oriane Plantex said her mic doesn't work, but she said she was wondering if you received any political feedback or regards. Is there any political feedbacks from your work? From
not yet,
decision makers.
I think that the databases we built, and especially here in São Paulo, we're under the umbrella of the Biota project in FAPESP. Carlos Joly is here present. He coordinates that for a long time. And so mainly we're generating data and we're be generating more results, which will be fed into the BioAuthor project. And I imagine, I suppose, that that data also will be used for public policies and public policy, for public policy in general, by the São Paulo government or whatever, or regional governments. So I imagine we don't have a direct line yet, but once we start feeding the BioAuthor project databases with this data as well, we'll have a direct line with public policies. Regarding
for sure, for sure, the data is going to be used for improving public policies, at least in São Paulo state, if possible, in a larger scale as well.
Yeah. So the idea is that that's the idea of a BioAuthor project, is the idea of this funding, and we can surely build on that. So I know that BioAuthor project, I participate in many BioAuthor projects already, and I know that they have a very big database, especially on vegetation. So now bringing macrofauna and bringing in earthworm data, and not only for Atlantic forest, but also for the Amazon, I'm sure that will generate lots of information for better public policies in that sense.
Perfect. Well, thanks a lot. Again, thanks everyone for being with us. A special thanks to our speakers, Michael, George, and Jérôme. As I said at the start, at the introduction, stay tuned for the next Cesabinar at the end of May, on Thursday, 28th of May. It will be about tropical forests, but we will look at the intraspecific variability of 3 species. And, have a good afternoon or morning, depending on where you are. Bye-bye. Cheers,
Bye-bye.
everyone. Thanks.
Thank you very much.
Thank you.